Hi, I'm Michael,
And I'm Akash.
and we're both members of Team Change. Today we'll be presenting on gene targeting techniques for Huntington's disease. Huntington's disease is an autosomal dominant neurodegenerative disease. It's caused by an extended CAG trinucleotide repeat in the HTT gene. This mutation causes symptoms affecting memory, motor control, mood, and more. These symptoms are primarily caused by the mutant HTT protein aggregating and accumulating in neurons. In this presentation, we'll look at various approaches to reduce the expression of this mutant protein. These include RNA-based methods, as well as allele-specific and non-allele-specific DNA-based methods. I'll hand it off to Akash to go over methods with you.
Perfect. So in terms of our methodology, there were four main areas we focused on: RNA targeting, DNA targeting, allele specificity, and disease models. For RNA targeting, we then divided it up into three other sections: one based on small interfering RNA, a second on microRNA, and a third on antisense oligonucleotide-based approach. The main goal of all of these approaches is to determine how each work to reduce the production of toxic mutant mRNA transcripts consisting of CAG repeats of Huntington's disease. For DNA targeting, we delved into CRISPR-Cas9 genetic engineering approach. For allele specificity, we delved into targeting endogenous SNPs and established Huntington's disease haplogroups. And then finally, for disease models, reviewed different cell model types that are crucial for studying and modeling Huntington's disease. Now I'll pass it back on to Michael to discuss the results.
So various different RNA-based approaches have shown promising results in different studies. In a study by Boudreau et al., an artificial viral-mediated microRNA resulted in improved behavior and survival rates in mice. In a paper by Stanek et al., an adeno-associated virus was used with a short interfering RNA to reduce HTT levels by 40% and improve behavior. An antisense oligonucleotide approach by Dadson et al. Reduced mutant HTT transcripts by 83% and wild type by only 43%. This technique displayed off-targeting effects, however, in other CAG repeat genes, but it did restore neurological function in mice. I'll pass it back to Akash for DNA targeting results.
Yeah, so for DNA targeting, we focused on two approaches: non-allele specific and allele specific. For non-allele specific, we found from the Zhu paper that Huntington's disease patients derived induced pluripotent stem cells were induced with CRISPR-Cas9, and effects were that the cells were able to be differentiated into GABAergic neurons, which had no signs of Huntington protein aggregates formed. For allele-specific approach, we found from the Shin paper that excision of the promoter region, transcriptional start site, and CAG repeats resulted in a healthy allele being unaffected. Then SNPs of the HTT exon 1 were identified to either create or destroy a PAM sequence on the mutant allele. The Shin paper found that about 80% of the HTT fibroblasts had a specific SNP in the promoter region, which resulted in elimination of the PAM sequence. This was then used to help target and delete the promoter on the mutant allele. Now I pass it on to Michael to discuss conclusion.
So both DNA and RNA approaches have shown significant promise. RNA approaches have been more thoroughly researched, but with recent advances in CRISPR technology, it may become advantageous for permanently decreasing the expression of mutant HTT protein. Off-targeting is one of the main risks of these techniques. Some approaches have directly targeted the CAG repeat, but this has risks because of other CAG repeat genes, increasing the off-targeting chances. Instead, targeting of SNPs allows for allele specificity, which avoids targeting the healthy allele. For this SNP-based approach, individual screening of SNPs would be needed. Lastly, I'll hand it back to Akash to go over future directions of research.
Yeah, so for our future directions, we aim to utilize the gene editing technique called prime editing to target mutant Huntington gene by inserting stop codons that would help to induce an early termination to the production of mutant Huntington protein. This approach significantly reduces off-target effects that are limitations from CRISPR and potentially can increase gene targeting efficiency. This is the end of our presentation. We hope you enjoyed it.